Welcome to the Performance Around the Clock podcast. I am Professor Sachin Panda, a scientist in the field of circadian rhythm. In this podcast, I will talk to experts in many different fields about science, health, and human performance. You'll also find out why circadian rhythms is my favorite topic. I hope you enjoy the podcast and thank you for listening. Welcome to another episode of Performance Around the Clock. And today we are recording from Lubeck, Germany. And I'm super excited to have Professor Anna Krook from the Karolinska Institutet in Stockholm, who is an internationally recognized leader in metabolism, specifically insulin regulation or insulin resistance, insulin sensitivity. And since insulin's action is mostly through muscle, that also brings us to muscle function. So she has done some pioneering work in the management of type 2 diabetes as well as exercise. And both these topics are dear and near to my heart. So welcome, Professor Anna Krook.
Thank you.
Yeah, so, when people hear your name these days, they always relate to this morning versus afternoon exercise among type 2 diabetes. But I'm sure there was a long research career leading up to that work, and we'll get to that work. But I'm curious, how did you get into insulin and muscle?
Well, insulin and the insulin receptor was the subject of my PhD work. And so together with now Sir and Professor Steve O'Reilly, I looked at the insulin receptor gene in people who had really, really severe forms of inherited insulin resistance. And we identified some really interesting mutations, stop codons of the insulin receptor. And so the next step was to try and understand, well, for the stop codon, of course, you have no insulin action, but for the other ones, to understand what was the implication in the insulin signaling cascade. But these mutations are extremely rare. And so I became really interested in a more sort of common dysregulation of insulin action, so people with type 2 diabetes. And as you just said, muscle is the main place where insulin action goes wrong, and is evident even before you get type 2 diabetes. So that became what I was really interested in. So that's why I followed the insulin signal into the muscle.
Mm-hmm. Yeah, let's wind back because many of our listeners may not know what is insulin resistance and why it is important to study. And when you say genetic form of insulin resistance, then how does it look like? How parents can figure out whether their kids have a defective gene? And you said it's very rare. So can you disentangle some of these?
Yes. No. So these syndromes are very, very unusual in that you have a mutation in the actual gene that makes the insulin receptor. So for insulin to work, it comes along in the bloodstream and it has to find a receptor. And in these very rare situations, the insulin receptor perhaps has mutated so that insulin can't bind to it anymore, or insulin can bind, but then the receptor sits through the membrane, so it has little signals to the inside. Maybe those parts of the receptor aren't working. Now, we have a gene from our mother and our father. And in most cases, even if you have a bad mutation from one parent, the other gene that isn't mutated will compensate. So in order for you to have these very rare syndromes of insulin resistance, you have to basically have a mutation that comes from both your mother and your father.
Mm-hmm.
So that's one reason why they're very unusual. And they might be slightly more common in places where consanguinity, so when it's more normal that cousins would marry, you could see that. But in general, they are very, very rare.
So it's almost like the insulin is the key that comes and latches onto the lock, that's the insulin receptor. And if it fits in and wiggles and the lock opens, then that door is open for the glucose to go from outside to inside the cell.
Exactly.
Yeah.
And glucose is a small molecule, but it can't just get into the cell by itself.
Yeah.
So just like you say, insulin has to bind to its receptor, then several things have to happen, and then the door opens.
Yeah.
And glucose can come in.
And why muscles are so addicted to glucose, or why muscles take up so much of glucose? We always, people always say that your brain weighs only a small fraction of your body, but it takes 10 or 20% of glucose. How does that compare with muscle?
So for one, even the brain is important, it probably only weighs about kilo-ish, whereas your muscle, depending a bit on your body type, will take up almost half of your body weight. So just by that sheer volume. But muscle, and we have to give fat cell its due, is a little bit special in that our brain, the glucose door is always open because brain just loves glucose. So that door is always open, it never shuts. Whereas for muscle, the door only opens when insulin is there. When insulin is not there, or we will talk about exercise later, but basically, in the absence of some kind of signal saying take up glucose, muscle won't take up glucose, or very low levels of glucose.
Huh.
Because it has a specific glucose transporter that has to move from inside the cell to sit on the plasma membrane.
Yeah. Yeah, so so that's when you went from insulin receptor mutation studies to muscle. And I think one of the key papers I remember you did with Julene was to show that in diabetes, type 2 diabetes, the circadian rhythm in the muscle is disrupted. Now, you also said that insulin gets into the muscle, glucose gets into the muscle only in the presence of insulin. And what is the role of a circadian clock in this complex play of insulin, glucose, and muscle?
Well, I think that's a really interesting question because, as you have shown very nicely, a circadian clock helps us to anticipate what we need to do. And so one would wonder, what is it about the insulin and glucose metabolism that we need to anticipate? And we are mammals that are active during the day. So you imagine that we would be able to metabolize more efficiently, perhaps, during the day. And I think there are some really nice studies, not from our lab, but from Patrick Stroud's lab, showing that this ability of, and now it's not glucose, but the ability of the mitochondrion to really produce energy seems to peak during the afternoon in muscle.
So let's repeat that. So the mitochondria in muscle are more effective to produce power.
Yes.
In the afternoon.
In the afternoon. And we can wonder kind of why this is the case. And our evolution we didn't evolve to sit in lecture halls for 8 hours during the day. So perhaps in evolution, it somehow was more important that we had maximum power output in the afternoon. And I've thought a lot about this in the sense that we have all these circadian transcripts that we talked about. But circadian transcripts, and this was mentioned in one of the talks, but are only relevant if it comes out in some function. And so when we measure mitochondria, it's very clear that there's a functional rhythm to the mitochondria. And you kind of I really been wondering about, why are they not just there the whole time? Why do they live this life where they fuse and they separate, and then they have this maximum ability of producing energy in the afternoon? I don't know the answer to this, but it's clear that they really do. And the fascinating thing is that, for reasons that I also don't know the answer to, is that if you take muscle from people who have type 2 diabetes and you study this in culture, and the only special thing about the muscle was that it once came from a person with type 2 diabetes, then this rhythm seems to have disappeared. Or maybe it was never there. Of course, we don't know that, because we can't retroactively go back and study somebody before they get diabetes and know that they will get it.
Nice. Yeah.
But my assumption is that it was somehow induced by the impaired metabolism in the person, and then the rhythm is gone.
No, actually, I was thinking about that, why the mitochondria should produce more power in the late afternoon. And it reminded me of my 3 or 4 days of camping in Masai Mara. And I remember one day at the camping site, the rule was this: there was a strict rule that you must be back in your tent or hut or whatever it is before sunset, because there are wild animals outside. And means I'm very good in disobeying rules sometimes, and then I just strayed a little away to look at some animals, maybe deers or something, and then I lost my way. And all of a sudden, there is this very primordial fear that maybe there is a lion behind that bush. And I could feel that fight or flight kind of response, where I had to run or find my way before it's too dark.
Mm-hmm.
And so I think so that I can connect with another study that Horacio de la Iglesia from Seattle, and actually we have an episode with Horacio, and he collected this activity data from Argentinian Toba. And the Tobas are hunter-gatherers. They go out and they come back in the evening. And I was analyzing this activity data, and what I noticed was in the morning they would get out, and the activity, like the Fitbit kind of activity tracker, the step counts would go up, kind of stay steady throughout the day. And just before evening, there's a large spike. So maybe we're designed in sunset 150 years ago was a real hard stop. And if you're outside, and that in those times there was no, it's not that people can turn on the light on their phone and then come back home. So you have to rush back. Maybe that's the reason why we have to increase our power output and run back home. That's what I'm thinking.
Or I guess also that's when the animals are moving in dusk. So that's when you can
outrun
efficiently hunt them. Or also to get the food. I was thinking when of humans as predators.
Yeah, that's also true, because if you have a fresh kill in the late afternoon, and typically the family or the community or the tribe would light a fire in the evening. They're not lighting a fire at noon to kind of grill whatever you have killed.
No.
And yeah, so I never thought about the hunting aspect, because I was fearful of being hunted. So what you're saying is, even if you took, and you do this muscle biopsy a lot, can you tell us how do you do the muscle biopsy, and then what do you do to the muscle? Because a lot of people, they kind of cringe when they think that there is muscle biopsy. People are going to take a chunk out of their muscle.
It's not a big chunk, it's a little chunk. So we take muscle biopsies from the thigh muscle because it's a big muscle, it's easy to get to. And basically, and it's under anesthetic, in that you get a local anesthetic, and then you go in with a specialized little tong, and it's quite a quick procedure. So it's not, it is painful. You do feel it.
But you don't suffer from it.
But you don't suffer from it. We have taken muscle biopsies before and after a marathon on the same person. So they muscle biopsy and they run the marathon, then we take another one just to look at exercise effects. But
so wait, so you take a muscle biopsy, and then the person runs 23 miles or whatever, and then again you take a muscle biopsy.
Yes.
Did that person win?
No, but we had some anecdotal that some people said when they were feeling that that the marathon just wasn't going their way, and you hitting that wall, they was felt, but I'm part of a research project, I must run.
Okay, that's good.
So there was some extra motivation.
So you can take muscle biopsy from
and so we study the muscle biopsy a lot to see what's happening, but also from that muscle biopsy, in between, so muscle has a lot long fibers, and the actual muscle cell goes from tendon to tendon. So for example, when you talk about the thigh, it goes from your hip to your knee.
So these cells are really almost 1 foot, 30,
Yeah,
40
they are
centimeter, depending on
really-
how long is your leg.
really, really long, and they have loads of different nuclei, but you can't grow them. Those cells cannot be grown in the lab. They terminally differentiate, but we can study the snapshot. But in between these cells are something called satellite cells, and that's the stem cells of the muscle. And these are the cells that are activated if you have a muscle injury. They become activated and fuse and help to mend your muscle. Or when you work out in the gym to grow your muscle, the muscle hypertrophy. So these cells can grow. And so these are the ones that we sort of tease them out from between the fibers, we liberate them, and then we grow these on plastic. And so they have never actually, in their life as a cell, they've never been a muscle, but they have the potential to become a muscle. And so if you do a few tweaks to them, they can fuse and align, and they can do what the real muscle does, is that all the nuclei can come together and they share long myotubes. But they never become like real muscle. But they really are a good tool for us to then study. For example, if we find some really interesting gene that we think this gene probably regulates glucose metabolism, we can go to our muscle cultures, and those we can manipulate, and we can take away the gene, or we can overexpress the gene and then see what would happen. Mm-hmm. And especially for genes that are really specific to humans, then we feel that this is a better system than perhaps other cultured systems.
Yeah, but when you take the muscle biopsy, right away, you're doing some studies. You are actually studying, because most of the cells, or most of the mass of this muscle is still muscle cells. So the experiments you do right away within an hour or 2 or 3 of taking the muscle, you're mostly measuring the muscle cell or muscle nuclei, and then only after you put them into culture discs, maybe after a day or 2, the remaining cells that are alive, those are the stem cells or satellite cells, right?
Yes.
Yeah. So when you, going back to diabetes, and so let's go back to mitochondria, because we started the conversation with mitochondria. So if we take, let's go back to very basic, like a sedentary person like me, you take a muscle biopsy, how do you measure mitochondria function? Because people always say, well, this is mitochondria, powerhouse, and all that stuff. But after taking the cells out, how do you figure out whether the mitochondria are healthy or not, and how much power they're producing?
So I guess in theory, you'd want to measure ATP. We don't do that. What we do is we utilize these technology called the seahorse analyzer, which basically you have your cells growing on a little plate, and then you give them different substrates, and you see how much oxygen that they need, because mitochondria are dependent on oxygen. They are what breathes. And you can actually measure that in real time. It's really quite fascinating that you can see exactly what the mitochondria is using and how it's the oxygen consumption rate.
Yeah, it's almost like a bomb going off. So for the fire to light, you need oxygen or the air, and you are just measuring how much air or oxygen the mitochondria use. So that's a proxy for how much power it's producing.
Yes, which is...
And by the way, seahorse is not the real seahorse, it's the name of the company.
Yeah, it's not a seahorse. It's a company. Yeah. It has a cute seahorse as its emblem. But for example, if we didn't do the cells, but we just took the biopsies, we can use, it's sort of a fancy Clark electrode, where you can also just put in the whole muscle tissue, and you can see how it breathes in real time, which it's one of these. So if you imagine you're in a lab and you actually see the traces like on TV. Yeah. That's kind of, I don't know, I'm childishly excited when I see that. Because you actually see, the little, well, of course you don't see the mitochondria, but you see the output of them, and the effect of them breathing.
So going back to my stupid analogy of a bomb or fire, so then what you were saying was when you add substrate, you add a little bit of sugar or fat, and see how much the muscle or the mitochondria is breathing air. So how does it look like from a sedentary person like me and then from an athlete? Is there a difference or the mitochondria are the same?
Well, the an athlete will have, especially an endurance athlete, will have a lot more mitochondria per milligram of muscle tissue. So they will just require a lot more oxygen. So there's one of the things that just by exercise training, one of the things that you do is you send a signal to your muscle, make more mitochondria. So a very well-trained athlete will have orders of magnitude more mitochondria than a really sedentary person.
Orders of magnitude, like
Well, at least 10, so
at least
tenfold.
tenfold more mitochondria than a
But
sedentary person.
I mean, that's in the extremes. It's sort of in between. You can have like twice as much or
Yeah.
3 times as
It's
much.
like between me and somebody who got a gold medal in, say, Olympic marathon. Yeah.
And it differs a little bit what if in red or white muscle.
Yeah.
And the reason that muscle looks red is that it has more mitochondria.
I see.
So you can
Not because it has more blood.
No, not because it has more blood.
Yeah,
It has,
no, it's good to know because most of the time we connect redness in a meat with blood,
yes.
but actually redness in a muscle piece relates to mitochondria.
And so you can actually, for example, in chicken, you can sort of see the darker muscle and the lighter muscle, and that's also reflects differences in mitochondria. When I tell my children this, when they were small, they were like, "Oh, don't say that. We don't want to know these things."
We just want to eat them.
Yes.
So as you said, there is a big difference between a muscle from a sedentary person and muscle from a trained athlete in terms of how much oxygen, how much mitochondria, and how much oxygen they consume. Is there difference like that between a person with type 2 diabetes and person with healthy person?
So this is when it becomes really interesting. Most of the time we spend a lot of time trying to really standardize when we compare people with type 2 diabetes versus people who have normal glucose tolerance. We try and make sure that we have the same sex, we have the same age, we really try and have the same body mass index. It's quite hard to get the same body composition, but and even if you standardize as much as you can, most of the time you will find that somebody with type 2 diabetes tends to be less physically fit. And of course, you can normalize that away by making sure that you basically take the absolutely most sedentary normal glucose tolerance and match them with the type 2 diabetics. I actually think that somewhere, maybe that is part of maybe it's not part of the disease, but part of what would predispose you to becoming someone with type 2 diabetes. Perhaps you just genetically have slightly lower physical fitness. But even when we match them, we do see that there is this impairment in the muscles' ability to respond to insulin, and then this dampened rhythm. Or actually, I mean, statistically speaking, there is no rhythm in our cells from the people with type 2 diabetes.
So when you say no rhythm in the mitochondria function?
No rhythm in the mitochondria function. They had rhythm in other things, like if you just measure the mRNA of different genes, there is rhythm.
Mm-hmm.
It's just different from the cells that came from people with normal glucose tolerance. But when it came to the mitochondria, where we have this functional readout across 24 hours, then we have a rhythm in the cells that came from people with normal glucose tolerance, and we have no rhythm.
So when you're talking about normal healthy person, so the mitochondria, and you are sampling at different time of the day, the afternoon mitochondria are producing or using more oxygen, so they're more active. And the type 2 diabetic muscle samples, when you say they are flat, are they always producing more oxygen, or they're dampened so they cannot use oxygen?
It's dampened. I think
they cannot use oxygen much.
They just don't seem to be as efficient.
So it's almost like the type 2 diabetic patients do not have a circadian rhythm in their mitochondria. The powerhouse is kind of off, and it turns off most of the time, doesn't turn on in the late afternoon, as is found in normal person.
No, yes. And then, of course, we can discuss this as a cause or consequence.
Yeah.
We don't know that. And one always has to be critical about sort of the conclusions that you draw. So there are people who are born with mitochondrial myopathies. So basically they have a mutation in their mitochondria and it just doesn't work properly. And they sometimes get type 2 diabetes, but it's not from muscle insulin resistance that it develops. They normally have impairment in their pancreas.
Okay.
So you can actually have reasonably well-functioning muscle even when the muscle mitochondria seem to have, or we know that they have a mutation because you can, of course, measure that.
I see. So
But then, for example, we have electron micrographs of on mitochondria. It's the prettiest little organelle when you look at an electron microscopy. But the prettiness, they're not as pretty in the people with type 2 diabetes. They just look less happy.
That's
very, very subjective because you can't measure happiness in a mitochondria, but they aren't as structured in their cristae. They look kind of clumped together.
I see. So structurally, they are kind of damaged mitochondria. They almost look like they're...
In my very biased way of looking at it.
Yeah, I mean, it's very hard to describe certain things. For example, a healthy, nice leaf versus a crinkly leaf. So
it's sort of a bit like that. Like somebody took the mitochondria, squashed it up, and put it back in the muscle.
Yeah, so now let's go back to our previous conversation that a healthy person's mitochondria who is sedentary like me versus a trained athlete. So now we have a type 2 diabetic person whose mitochondria is looking very sad and also cannot consume too much oxygen, cannot produce too much power in late afternoon. That rhythm is dampened. Can we train that type 2 diabetic patient to make the mitochondria healthier?
I think this is what we need to do. This is the research that's sort of the next couple of years. We will probably do that. We know, though, that exercise can be really beneficial for people with type 2 diabetes. You can improve your insulin sensitivity, you can improve your overall metabolism. And the earlier in the development of the disease that you do the exercise, the more you're able to make your disease revert. Maybe it's hard to know whether you make it revert or you postpone it. But even if you postpone your type 2 diabetes for 10 years or 15 years, a lot is gained by that. So we do know that exercise works well in that way. Whether it restores rhythmicity, we don't know.
Mm-hmm. But it means we know that exercise increases the number of mitochondria in muscle. So similarly, has anybody done the experiment, even in mice, type 2 diabetic mice? Does exercise in type 2 diabetic mice increase the number of mitochondria in muscle? There must be something.
I mean, the exercise, so type 2 diabetes is a very human disease because we only have mimics of it in mice.
Yeah, I see.
And but if you exercise a mouse, even if the mouse is on a high-fat diet and it's insulin resistant, you will, first of all, you will ameliorate your insulin resistance, and yes, they will get more mitochondria.
They will get more mitochondria. So hopefully in humans also, they might see some improvement in either the number of mitochondria or the function of the mitochondria, because even...
Oh yeah, no, I think, and certainly exercise is well known to be able, even when you are resistant to insulin, for example, or if you have no insulin, like a type 1 diabetic, exercise will certainly activate, exercise-stimulated pathways will be activated, your muscle will adapt, you can get more mitochondria. whether you do it as efficiently, especially with people with type 2 diabetes, is it also a form of exercise resistance? That's a huge debate in the field.
I see. So, but coming back to this point, because this is what you said, type 1 diabetic who do not produce insulin and are dependent on insulin, when they exercise, they can also, the muscle can soak up glucose like a sponge, almost like a sponge. How does that work? Is it because they're not producing insulin, so insulin is not coming to open the door? And as you said from the very beginning, muscle is one of the places where you have to unlock the door. So since the key is missing, how is glucose getting into muscle? Do we know that?
We know quite a lot about that, and it's been studied quite intensely, because it's almost like there's a back door or an extra key, and that is muscle. Just the act of contracting the muscle will also send a signal to the same, or we don't know whether it's exactly the same, but it's the same protein. Maybe it was lying in a different compartment, but the same little glucose transporter will come and open the door for glucose. And if you think about it, it makes sense, because if say you get up in the morning and you decide you don't even have breakfast, you just decide you want to go for a run. Now your insulin levels are going to be pretty low because you don't have any glucose in your bloodstream, but you start to exercise, and basically your liver will then kick in and produce glucose to send glucose to your muscle, and your adrenaline will kick in, and muscle then, the contracting muscle, then opens the door and can take up glucose. And we know that there's one molecule that's really important for that, is the AMP kinase. And AMP kinase, it sits in the muscle and basically senses whether there is low energy level. And when AMP kinase is activated, it knows that we need more energy. Let's open the glucose door.
I see. So then we are coming to, you mentioned the person getting up in the morning and running. And as we said from the very beginning, your name always comes up when people talk about the person running in the morning and running in the afternoon. And there's this famous study, and maybe you can describe much better. What did he do with this? They were not healthy people, they were type 2 diabetic people.
No, so we started out with a study with basically was type 2 diabetic men that we recruited, and we asked them to do this thing called high intensity interval training every second day. And we picked this because we knew that it was a very efficient way of training for a fairly short period of time. It's only 30 minutes of training every second day, but we knew that it really would improve your muscle performance and all these things we talked about, like increasing your mitochondria. And this had been really published at that point to be shown that it's very efficient, more bang for your buck for your exercise.
So let's go back to high intensity interval training. For many of people who may not know, this is sitting on a bicycle and cranking to your,
So basically it was done on a bike, it was supervised exercise. Basically you exercise as hard as you can for 1 minute, and then you have a bit of a rest, and then you do a really hard exercise. You pedal for your life, and then you do this several times, and that's kind of the interval part.
I see.
So it's the high intensity interval. It's quite hard. I mean, it's not something that you just tootle along and do. So it's quite intensive exercise. And we then made use of these, what you can see people wear now. They have these little white patches on them where you can measure glucose constantly. And doing this, we were thinking that our initial hypothesis was that we'd have different chronotypes, and some people would really benefit from exercising in the morning, and some people would benefit more from exercising in the afternoon. And what then really surprised us when we got the results was that for the people who'd exercised in the morning, their blood glucose control, their curve for the whole day, because we could see that, was higher. They had just higher blood glucose that whole day and the whole next day.
Next day too, when they're not exercising.
When they weren't even exercising, just the fact that they got up early on a Monday morning and exercised, then their blood glucose was high all that Monday and the Tuesday.
So did they have to really wake up earlier than when they have to wake up? Is that?
That was, so we did, so the study ran that they basically did for 2 weeks. So they exercised Monday, Wednesday, Friday, Monday, Wednesday, Friday. And if we looked at the second Monday, Wednesday, Friday, then it wasn't quite as bad. They started to become a bit more sort of like they hadn't exercised, but it was in contrast to if they'd exercised in the afternoon, because then we had all these effects that we thought we would have, that if you exercise in the afternoon, the next day you had lower blood glucose levels. So there was this legacy effect of you've done this exercise, you had better blood glucose control, all these things that the field really thinks this is what happens when you exercise. And that's when we began to think that, first, we had all sorts of theories. Maybe it was the stress of getting up in the morning. We're still thinking that it could also be the fact that it's really quite an intense exercise program. This high intensity exercise, the people were previously sedentary, and maybe if they trained, maybe we ran it for a month, perhaps the morning exercise would become beneficial. Maybe you'd adapt to it.
But even this one is very interesting in the sense, just before this, we discussed how type 2 diabetic patients do not have a circadian rhythm in our mitochondria power production. So that means whether they're exercising in the morning or evening, they should actually have similar benefit because they might be cranking up in the morning versus evening. So that means although there is no circadian rhythm in mitochondria function, there is something else that's still sensing the pace of time. So it knows that this is afternoon, and this is when, if the patient is running or doing high intensity exercise, then there should be better glucose control.
Right? No, and I think this is one of the things. Of course, it's beautiful to work in a cell culture system where you have your muscle cells in a little dish because you can control everything. In vivo, though, there's a huge discussion between different organs. Your liver is giving you glucose, your fat cells is giving you lipids so that you can perform your exercise. And so there's this integrated system in vivo. And I'm absolutely convinced that people with type 2 diabetes still have a diurnal rhythm, as in that they probably feel sleepy at night. So I think that there is clearly a metabolic problem with the muscle that I think might have some sort of circadian misalignment. But at the same time, it's still better to exercise in the afternoon. And then the question, of course, is what about the healthy people, the normal glucose tolerant people? Well, for them, it didn't matter. Even though that they have this nice rhythm when I take the cells out and put it in a dish, for them, it was also because of the fact that if you have normal glucose tolerance, there is very little to correct. So we can't really expect to see any differences. But we went back and then did an extra experiment where we had people without diabetes. And it turns out that for them, it didn't matter.
But you raised a good point that these are already healthy, and we cannot make healthy to super healthy.
No.
Because, for example, so this also brings up another point. When we think of blood glucose level and what is considered healthy, in terms of, the Europe uses a different way to say that, and the US uses a different way. And I like the US way because it's very simple. 100 milligram in 100 milliliter of blood, although I don't know how that happened. For some reason, when US is tied to gallon and pint and ounces, somehow they got 100 milligram per 100 deciliter, 1 deciliter. So now we also know that there are some people whose fasting blood glucose is around 90, 95, and you look at that chart for the last 10 years, they're still at 90, 95. And then there are some people whose fasting blood glucose may be between 80 to 85. Okay, so there is a 10-point difference now. So the point is, can you take the people who are 90 to 95, make them exercise in the morning versus evening? Will they go down to 80 to 85? And I think those kind of experiments will be really interesting to see whether there is a set point that you cannot change. But once they go outside the set point and they're going from 100 milligram per 100 ml to 125 or higher, which is type 2 diabetic, only there you have an effect of exercise. But when you're healthy, then it doesn't make much sense to go from healthy to super healthy.
Well, I think, though, that even in the healthy people, you will have exercise benefits, but you won't see it so clearly. You might
The
be
morning
right.
versus evening exercise.
But maybe not in the morning versus, but maybe you won't see too much on the actual glucose values either. But what you will see is that you will need a lot less insulin.
I see.
So the amount of insulin needed for you to maintain that.
So that you can actually preserve your pancreas for a longer time. So, okay, so that's why people should exercise to delay the potential onset of type 2 diabetes or pre-diabetes.
Yes.
So now you work in this area a lot, and we know that, for an 18-year-old, the chance of being diagnosed with pre-diabetes is pretty low, almost negligible. As we get older, then we are more likely to be pre-diabetic. Of course, you work in Sweden where people are very active, but still in Sweden, do you have any epidemiological data like what is the prevalence of pre-diabetes or type 2 diabetes among people who are 60-year-old or 80-year-old?
Well, I think you're right. It goes up with age.
Yeah.
Clearly, it goes up with age. And presumably, if we live to be 120, maybe we'd all become
Pre-diabetic or
pre-diabetic or diabetic as our beta cells, just the cells that give us insulin to keep our blood glucose down, just probably get a bit old and tired.
So then exercise...
But you can delay it, I think, by lifestyle. And I think that's been super clear that the things like physical activity or diet really can push or go the other way. It can walk down in age on when you develop type 2 diabetes.
Mm-hmm. So exercise is almost like your investment into future. It's your retirement fund. The more you save, then you'll have a more comfortable life. So this is for average people. So now what about athletes? If they exercise, or weekend athletes, because it's really hard to work with Olympic level athletes or weekend athletes, will the benefit if they exercise in the morning versus evening? And when I say benefit, there are many different ways to describe what is benefit. Because somebody might, some people will go for exercise and then come back and say, well, I feel great. They might have done the same time, like they might have gone 10 kilometers in the same time, but sometimes they come back and say, I feel great, because they're not exhausted. And is there a difference when weekend athletes do this morning versus evening exercise? And when we say morning, maybe it will be better to define what is morning. Is it getting up at 5 o'clock or 6 o'clock or 7 o'clock? And
yeah. So this is the study that we're doing at the very moment, where we're recruiting people who are pretty intense about their cycling. They're quite good at cycling, and they're pretty intense and interested in this, and they train for 3 to 4 days per week. And we have then tested them at 9 or at 6, in 9 a.m. or 6 p.m. And we have a few people, different chronotypes, and asked them whether they like to exercise in the morning or the afternoon. And just on the very preliminary data we have is that we were astounded in that when the performance as measured by a 10-kilometer time trial on a stationary bike, it was identical. Completely to the second.
So these are the people who did a morning exercise for how many days or weeks?
So they train pretty intensely. So they train 3
to 4
days per week. They probably sign up for these cycling races, and they train for those. And so they're not athletes. They're not professional athletes.
They're not doing too
well-trained young people in Stockholm.
Yeah. And so when they do that in the morning, and they did that in the morning for a few days, and then they switched over the afternoon. That's what
No,
they did?
So this is just basically just test what your hypothesis is. There, if we just bring them in one time in the morning and one time in the afternoon,
Oh, I see.
will they perform better? the afternoon, perhaps, when they have more mitochondria. And it turned out that, no, they were just spot on identical.
Wow.
Even,
There is no difference, like even to 0.01 second or anything?
Well, 0.1 second, yes, but within a second. So the average for these 10 people was 1,070 for the morning and 1,071 for the afternoon.
Oh, so that took longer time in the afternoon.
To a second. It wasn't statistically significant, this.
But it's like...
The difference is 0.0093%.
Yeah, but then in a race, that 1 second would have...
True.
But the afternoon, in the afternoon, they actually went worse.
Yes. Not all of them. So there was some that went a bit high. So within, I think, speaking to my colleagues in the lab, I think the largest difference they had was a 4-second difference.
Largest difference was 4 seconds. Yeah.
But most were within a second. And they went back to analyze, was it then a familiarization in the sense that, did it matter whether it was the morning that was the first time they were in the lab or the afternoon? And no, there was no difference there either. There was also about a second difference if you just said morning versus first time versus second time, irrespective of morning and afternoon.
So this was a time trial. So they were really pushing hard to get
it was a time trial.
These athletes are really highly reproducible, because when I run my 9-kilometer run, difference can be minutes.
No, and I think kudos to the guys running the study, because they really have to standardize everything and kind of the exact power and how they've done this. But I think this is the beauty of science. We went in to do this experiment thinking this is what we'll find. We do the experiment to the absolute best of our ability, and we get a slightly different result. So that means we have to go back and think again.
Yeah, so that's mind-boggling, because everybody is talking about afternoon exercise being better, and even there are studies now going back to Olympic time for various competition and showing that no, the afternoon timing is much better and all that stuff. And then when you really do a well-controlled study in the lab, then you see a very different result. And I was surprised that the difference was less, only 1 second in this 10-kilometer. Like,
yeah, yes, we haven't quite digested that either.
Yeah,
but I say it's the beauty of science. And I guess the other thing is that we've collated the data on 10 men. We are now also studying women. And it might be that it's different in women. We don't know that. And I think that's one of the things, that women are very understudied.
But you did this only, so they had to do this only once, right?
Yes.
What is the recovery? Are you measuring recovery to see whether they're recovering better or worse? People...
We don't know. Well, we do it within, because of the fact that we have set everything up so it's sort of within their max, sort of a percentage of their peak.
I see.
So we recruit them, and then we want to do the actual experiment within a week to 10 days after we've measured that. And then sort of visit 1 is sort of on a Monday, then visit 2 will be the following Monday, just to make sure that we, especially these people who are practicing for various races and things, we want to have them, catch them in that window so that they don't detrain or do extra exercise.
Did they know whether they're coming for, they would actually know, 2 or 3 days in advance, whether they're coming in the morning or afternoon?
Yes, so they do know, because we have to tell them so that they turn up.
Yeah,
it's hard to do a blind randomization on people like that. But we give them food for the day before, so that they've eaten exactly the same food, and of course, if it's the afternoon, then they have standardized food for the whole day. Otherwise, they just have standardized breakfast.
Yeah. No, that's really interesting, because I think the one experiment that I'm thinking is, of course, this will be difficult, because people will have, so suppose they come in the morning, you schedule all of them to be coming in the morning or in the afternoon, but only when after they come, then you tell them whether it's a time trial or they can take it leisurely.
Yeah, That we haven't tried, but I can tell you we have established cells from, we're taking biopsies, but cells take a few months to grow. So then we will be able to do this, see, do they have a better rhythm? Whatever better is.
Yeah.
How do we know that it's good to have a rhythm in your mitochondria? We're assuming that that's good.
Yeah, so there is a lot of assumption that it might be better, but we actually don't know. And then since it's the loss of rhythm correlates with type 2 diabetes, which is bad, then the question is the loss of rhythm causing, or the other way we can also say that type 2 diabetes is so bad that these mitochondria lose their rhythm to sustain health in type 2 diabetes.
It could be an adaptive response.
Yeah, so that's why sometimes, so sometimes we just correlate these two, saying that since type 2 diabetes is bad, the loss of rhythm is also bad, which may not be true. As you said, if we go back to doing the experiment, then we'll know. So what keeps you awake at night these days then? What is next?
Well, so there's loads of interesting things. One of the things that really fascinates, a few things keep me awake and that I'd love to do. One is that I'd really like to know why, when you exercise, does insulin work better? I just want to know why. It's the same person. You don't have to worry about different genetics or anything. It's like before and after exercise, and insulin works better. I just want to know that.
But
it's
how long?
Interesting that this is the thing that keeps me awake.
But how long does that effect last? Like suppose say I exercise today, and my body
hours.
Only hours.
Yes.
It's not a long-term effect.
It's not a long-term effect.
So that means I have to exercise every day.
Yes. So the analogy that I normally, when I speak to kind of the public, is it's a bit like brushing your teeth. You can't brush your teeth for an hour on a Sunday. You have to kind of keep doing it.
Yeah.
Exercise is a little bit the same.
I see.
Which it sort of depresses me, especially when we travel to conferences. And I go to bed and I think, I walked 3,000 steps today. How did that work?
So then what do you tell now type 2 diabetes patients? Because, another thing is you're doing this high intensity interval training. And when I asked you, can you please describe, that means a lot of people may not know and may not practice high intensity. A lot of type 2 diabetic patients may be just taking a stroll in the park. And what happens when you take a stroll in the park in the morning versus evening? Has anybody done that study?
No, and that's another study that we'd really like to do, because we have a suspicion that it could be an interaction between stress hormones and metabolic disease. One of the previous fellows in the lab, whose data I showed today, but who's now set up his own lab, he's actually looked at whether you take metformin, a common type 2 diabetes medication, before or after just going for sort of a walk, and that has different sort of the timing of the medication versus the timing of the walk. And so it might be that maybe if we tightened out the exercise to not be so stressful, maybe we would be beneficial also in the morning. So we don't know that.
So now you bring up another thing, metformin or drug. So those most type 2 diabetic patients who know that they have type 2 diabetes, there must be on some kind of drugs. So now the question is, have you studied whether exercise will further improve their insulin sensitivity? Well, if they're on metformin or if they're on GLP-1 receptor agonist, because the incretins are now so popular that people will forget metformin in 5 to 10 years.
Yeah, no, so the GLP-1s are interesting, and I think there's quite a lot of effort now going into, because they make you lose weight. Mm-hmm. And when you lose weight, you don't just lose the weight you want to lose, you also lose muscle mass, which you don't want to do. So there is a lot of research going on now as to how much exercise you'd have to do to mitigate the loss of muscle mass. But we haven't studied that yet. The people that we studied have taken their diabetic medication as well. Mm-hmm. And one can think about this in different ways, in the sense that that's their clinical reality. That's how they
Yeah.
live. So you could argue and say it was the metformin that killed the rhythmicity of the mitochondria. But of course, once we culture them, they're not in metformin anymore.
Yeah. No, I'm not concerned about
Yeah.
that. I'm saying, like you pointed out, now GLP-1 receptor agonist, and there is not much exercise studies on them so far, right?
No,
You are in that field, so maybe.
that's probably what we need to study now.
Yeah.
Because even during the last study that we did, we realized that a lot more, even people without type 2 diabetes, were now on the GLP-1 agonists. And we suddenly had to start to think about, should we include them in the study or not include them in the study?
Yeah, I mean, now I think the estimate is in the US, 1 in 4 type 2 diabetics is now on on incretin GLP-1 receptor agonist. And in 5 to 10 years, it will be almost 50+%. It's almost like the new statin. Like the rule of thumb is if you're 40 and a male in the US, then it's almost taken for granted that you must be taking statin. So similarly, I think it will be for GLP-1 receptor agonist.
Yeah.
And then the question is, what is the impact of GLP-1 receptor agonist directly on muscle, and then what people can do, whether they should be eating more or exercising. What is the optimum exercise? Because you also don't want them to exercise too much because they cannot eat. They cannot compensate for that exercise. They may not be able to ingest enough food to sustain their muscle mass. So I think that'll be an exciting area of research.
It will be. And I think we will start to see a lot more studies focused at maintaining muscle mass.
Yeah.
And like you say, what's the minimal amount of resistance training you need to do to counteract the effect of muscle loss from the GLP-1? And I think one of my clinical colleagues was discussing and saying that the real danger could be that if you go on the GLP-1 incretins and you lose a fair bit of weight and then you go off them, then it's well established now that the minute you stop taking this, you start putting weight back on, but you tend then not to put muscle back on. You tend to put fat back on. So then if you go back on the GLP, it's kind of like you slowly
keep losing your muscle mass.
more and more muscle mass and changing your body composition, which may not be good.
Yeah. So I think this gives us a lot of food for thought. And then maybe next time we meet, I expect to see a little bit more study from the healthy athletes, morning versus afternoon exercise, to see whether if there are other benefits or other adverse effects on time-dependent exercise. Because the bottom line is many athletes and many weekend athletes, they hear about the type 2 diabetes study and then they're trying to see whether they can do much, they can get more bang for their buck from the afternoon exercise. But at the same time, from these patients, you are also collecting a lot of other data, right? So you said that you did muscle biopsy, you collected blood samples, and are there any other differences for healthy people? Because for type 2 diabetic patients, I guess there are also other changes for morning versus evening exercise. What are the other changes in type 2 diabetics, and whether those changes happen in healthy people?
Yeah, no, so that's what we want to do now. So now we need to go back to the lab and we will analyze the metabolome and the lipidome and see, is that different? Is the response different in the people with type 2 diabetes? Is it different in the morning versus afternoon? The other thing that we've collected samples for is that there's this thing called extracellular vesicles. And to try and tease out, are they different in the morning and the afternoon? Are they affected by the exercise? And what their cargo and what they're doing. So there are all these things that we really try and tease out.
But in type 2 diabetes, morning versus evening, are there differences in inflammatory marker or stress? Have you looked at any of that?
Yeah, so one of the things that at least correlates with the morning exercise in type 2 diabetes is higher inflammatory markers and higher stress markers that are circulating in the plasma. And again, it's one of these things, it correlates nicely. And whether it's a cause or effect, we don't know.
So the morning exercisers will have higher level of
Higher stress levels and higher C-reactive protein.
Inflammatory
which is an
markers.
inflammatory marker, yeah.
But is it because of, so you're saying that you don't know whether it's because of exercise or whether it does cause or effect. That's what you are saying, right? But
and
that
is
also,
that affecting then the fact that the blood glucose levels are higher?
Oh, I see. So what you're saying is whether the morning exercise is causing the inflammation to go up and stress to go up, and that's
and
actually
that means
keeping
that your glucose isn't coming down like we would expect it.
I see. So it may not be a direct effect on the muscle, but some indirect effect through stress or inflammation. Yeah, that's a great point. And then if you take, say, trained athletes, and maybe these athletes are so trained that they may not show that the stress response or the inflammation response.
So once we've collected all the data from, because we want to analyze it in one big batch, that's what we want to see. And then we also have an effect of, we can also look at the effect of age.
Mm-hmm. These are all done in young people.
Because these are all young, between 20s and 30s.
I see.
The athletes, whereas the type 2 diabetic and their matched normal glucose tolerance, they are sort of in their late 50s.
I see. Yeah, so there is a big age difference here.
Yeah, so we can also try and see whether we see any obvious differences there.
Yeah.
And the sex difference, which we still have to kind of dive into.
Okay, so let's synthesize all of this. And, I'm in that 50 plus age club now. And there are a lot of listeners here who are also in that age club. After all of this research, would you say that people who are type 2 diabetic, if they do afternoon exercise, they will get better bang for their buck? Or should they forget about it and then just do exercise whenever they can?
I think that we can now have enough data saying that if you're going to do fairly intensive exercise, it is probably much better to do it in the afternoon.
Okay.
But in general, I would say exercise, and maybe what we didn't talk about so much, and which I haven't studied so much, but maintaining muscle mass is really important.
Mm-hmm. Okay, so because that's a clear message that many people want to hear. If they are type 2 diabetic, if they are at a risk for diabetes, then what they should do. And I think what you said, there are at least 3 major benefits. One is the blood glucose level will be much better if they do afternoon exercise. And the inflammation and stress hormone levels or stress markers may not go too high, because inflammation is also the cause for many other downstream diseases, the other diseases that can happen or that can exacerbate. And stress also affects other stuff. So the afternoon exercise it is.
Indeed. More afternoon exercise.
More afternoon exercise, but not too late in the afternoon or evening, because there was another study that came out from Australia analyzing tens of thousands of people and their sleep data showing that if you do evening exercise, then you are likely to sleep less. But then the question is, are they sleeping less? And that's sleeping less is really sleeping less, or they're getting so much quality sleep that they can get away with less sleep. So I got to ask that to those researchers. But until then, thank you so much. This is a lot of insight. And actually that 2019 paper is such a huge watershed moment in exercise physiology, because until then people thought that exercise at any time for type 2 diabetic is good, and that 2019 paper actually started all this new research on when we should exercise, young versus old, type 2 diabetic versus not to type 2 diabetic, and then now GLP-1 users and so on. Thank you so much, and I'm looking forward to talking to you again in a few years when you have all the data.
Thank you very much.
Thank you.